the two of you have allowed me to go ahead. So now I want to share digestion. Okay, so I can start now. I assume you're able to see my screen. So we are starting on digestion and absorption. And since we did some of these work in the pre-lab, I assume it will be a short lecture, unless you have questions. So we are looking at digestion of the mechanical chemical. I think I've talked a lot about this and the enzymes involved. So what we are going to do is to break the foods into the major categories. So we have carbohydrates, and the carbohydrates we mainly have polysaccharides. We have those that are found of plant origin, and there are others of animal origin. So mainly for the plant origin we have starch, but in animals the polysaccharide we normally have is glycogen. We also have things like the disaccharides, and I remember telling you when we were doing your test in the lab that don't always say when the polysaccharides break down they give maltose. Remember that they do give they can have maltose, but then when these ones break down they can have glucose, they can have sucrose, and they can have fructose. So you need to say generally whether they are disaccharides, monosaccharides, or simple sugars rather than being very specific. And now, generally, the plant uh, storage material that we have, we say is starch, and starch comes in form of aminos and aminopectin, and you find that it caters for about 60% of what we normally consume. In addition, we have a lift from glycogen, which is of the of animal origin. Now we have the rest coming from the sucrose, things like the sugar, the cane sugar and milk sugar. They account for very little. And in fact, as you grow older, you find that our dietary contribution from things like lactose we start reducing. Then we also have the monosaccharides, fructose, and glucose. This one usually you are given when you are really short of energy or you want to boost your energy, you go straight for the fructose or glucose. Then, of course, we have those other carbohydrates that we take in that are not digestible. These are the plant products, things like cellulose. You find that those ones they are not digested in humans and therefore they do not contribute to our nutrients. So what's the difference between aminos and aminopectin? If you look at it, it's simply the arrangement of the sugar molecules, where the aminos consist of mainly a linear helical chains of the glucose units. And if you look at where they are joined together, they allow this the 1,4 glycosidic bond allows for that linear arrangement, as opposed to aminopectin, which in addition to having this linear arrangement, in some parts you're going to find they have a 1 to 6 branch. Again, you need to understand some biochemistry, and for that I'll go to the next slide, which will show you the arrangement of the glucose monomers. So you have, this is the 1,4. If you look at the carbon numbers, the first one is number 1, and this one, if you count from number 1, 2, 3, 4, this is the fourth one. So 1,4 arrangement, glycosidic bond. Now, on the other hand, if it goes beyond that, that's 4, 5, 6, you have 6 position 6 here. So if it links between the 6 and the 1, then it is 1, 6. So it's the other way around, it's counting backwards. So it is 1, 6. Now, when you break down, because of this linear arrangement, you have the amylose. If you break this down, most likely you are going to have simple glucose units, which are monomers, or you can have disaccharides, which are 2 units, or you can have things like the trisaccharides, 3 units. When you start having 4 and more, then you're talking of short polysaccharide chains. On the other hand, when you have this one, the amylopectin, and you start breaking it down, it can also give rise to simple glucose units, or it can give rise to maltose units where you have the disaccharides. But it can also give rise to something with the 1,6 linkage, which we call limit dextrin, so that you have a chain here and a 1,6 bond and another chain here, which is what we call the limit dextrin. So these are the likely products when you start breaking all these glycosidic bonds. Now, in this diagram below here, I just put it to show you the relationship between the amylose and the amylopectin and glycogen. So if you look at amylose, it's just a linear chain. If you look at amylopectin, because of that 1,6 glycosidic bond, it has some branches. But the most branched of all is glycogen, which is found in animals. So you can see that it has a lot of these 1,4 linkages, but at the same time, it also has the 1,6 linkages, which allows for multiple branching. So this is the structure for glycogen, but all of them are made up of the simple glucose units. That means any of these, when they are broken down, finally they'll give rise to these units. Maltose, maltriose, when you have 3 of them, or you can have the limit dextrins. Now these products are the ones which will be exposed. Usually this breakdown of these chains occurs, it starts in the mouth due to exposure to salivary amylase. And the amylase is the one which will cut down all these long chains into these shorter units. Now these units will be within the chyne and they'll end up in the small intestines. And when they are exposed to the brush border usually has enzymes that are going to do the final digestion or the final breakages of these chains so that they release the individual glucose units or the individual monosaccharides. So as you can see in this slide, I have just said that initially you start in the salivary glands. So for carbohydrates, you go to the stomach. There is no digestion in the stomach. Then you go to the duodenum, where the pancreas is going to release the pancreatic amylase. And the pancreatic amylase will actually is going to cut down and release maltose units or the maltriose units or the limit dextrins. Now, once you have these, they are exposed to the brush border enzymes. The brush border enzymes you will have are going to break it. You have maltose, you have the enzyme maltase, and it will break these into 2 glucose units. In case you ingest sucrose and it has a glucose and a fructose unit, then you find that the brush border enzyme that you're going to have is sucrase. And when you have this sucrase, it will break it down into glucose and fructose. If you have the milk sugar, which is galactose, you have the lactose, it will break into the glucose and galactose using the lactase enzyme. So these enzymes, the sucrase, lactase, all those, they are found at the brush border, and they're the ones which are going to release the individual monomers. In most cases, only the monosaccharides will be absorbed. So you find that you hardly have the saccharides or any other larger molecules being taken in. But when they come to, they're exposed to the brush border, you release the monomers. Those are the ones that will be absorbed into the blood circulatory system. So having looked at all that, you've broken down your carbohydrates, and you have all these individual monomers. The monomers that you're likely to have is glucose, fructose, and galactose. How are they transported into the small intestine? So the next thing I will look at is the absorption of this. Now, the glucose and galactose, they usually use the same carrier, transport carrier. Then you have fructose has its own carrier. So among the three that I said are the end products, you find that these two will share the protein carrier and fructose will not. Now, I will come to the fatty acids later. So for now, I'll focus only on the carbohydrates. So as you can see, this is the diagram I had shown you at the beginning of the lectures, when I had given you the pre-lab showing you the end product of starch digestion. So hopefully, if you are to do the salivary test, you are supposed to be getting things like maltose and some glucose. But I understand you had some challenges with that. We are still looking into what the problem is. Now, the next step is absorption. So from this diagram, I have an absorptive enterocyte, that epithelial cell that is able to absorb nutrients. On the upper portion, we have the luminocyte of the small intestine. On the lower portion, we have the blood capillaries, which are on the basolateral side. So nutrients will be leaving the lumen and will be passing through the enterocyte. So the enterocyte is the one that determines what moves in, and then it releases it from the basolateral side. So absorption actually takes place in several steps. One, you have to have, it is an energy dependent step. Why? Because we have the sodium potassium pumps which kick out sodium, and when they kick out sodium, they bring in potassium, and what they generate is ATP, and this ATP is the one which will be used in other processes. Now, when you come to the luminocyte, the luminocyte uses concentration gradient. What we'll be doing, because now you have created a gradient, you have removed the sodium, and this sodium, once it goes into the PCF, it is taken away. So now what you have done is you've reduced the amount of sodium within the enterocyte. So the next thing that will happen is sodium from the lumen will want to go down its concentration gradient. And as it moves down, it uses a symporter. If you remember what is a symporter, it's a transport carrier which is transporting two molecules, where it is using the energy created by the gradient of one molecule. In this case, the gradient is that one of sodium. So we have low sodium concentration here and higher sodium concentration there in the lumen. So as the sodium binds, the next thing that will happen is the glucose will also bind to the same carrier protein, so that when it changes its shape and it exposes these two to the luminous, to the cytosolic side, it will release the sodium, and because of that, it will also release the glucose. So what it will have done, it will have transported sodium down the concentration gradient, and glucose is utilizing that to be transported into the cell. Now, it can either take the glucose or it can take galactose. So either way, it will take either of these two. Now, I said that these two share the same carrier, transport carrier, but fructose has its own. Yes, Asna?
Madam, can you repeat what you've said about the exchanger once more?
About the
exchanger.
This takes us back to the previous course where we were doing transport molecules. You remember that you may have a transport carrier. This is the cell membrane, and you have a transport carrier that can bind two molecules. So if it has a pocket there and it has another pocket here, or another, the other way around. Okay, let me just put that way. So it has a pocket for one molecule and a pocket for a second molecule. So what will happen is this first molecule, in this case we have sodium, will attach. And once it attaches, it allows for the second molecule, which is sugar, either glucose or galactose, will come and insert itself there. Now, once they insert themselves in that area, they will cause a change that will shift the whole of this side to the inside of the cytosolic side. Once it changes its shape and it exposes now the bound parts to the inside, now what you'll be having is something like this, where you have the sodium is on this side, and then you have the glucose in that position. So remember now the sodium is attached here and the glucose is here because it has shifted. So this is now the direction. So what will happen is it will release the sodium, and the minute you release the sodium, it also releases the glucose. So now you have released the two molecules into the cytosolic side. But if this transporter is not bound to these two molecules, it shifts back to that direction so that the receptor or the pockets are exposed to the extracellular surface. Are we together now?
Yes, thank you.
Okay. So that's how it ends up using the transport carrier for sodium. The glucose or galactose will be used as it's a symphoter. It is taking the two molecules in the same direction. So all this will be released into the cytoplasm. So now you have a high concentration of these molecules, the sugar or galactose. When you have all these, the next thing that will happen is that they'll use a transport carrier, really not a carrier, but it's like a channel, because all it will do is that it will use facilitated diffusion. Using this carrier, it will just be moving to the other side, leaving the cytoplasm and going into the extracellular side. So the carrier on the basolateral side is the same carrier for all the glucose, all the monomers, whether fructose, galactose, glucose, they are the same. And this is what we call the GLUT2 transporter. The other one is actually called a sodium galactose or sodium glucose transporter. So now the other one will be the fructose transporter. This one is sodium glucose or sodium galactose because it is a simple, it is using both to be transported. So on the basolateral side, what you have to keep in mind, we have a lot of sodium pumps, the sodium potassium channel, and they create energy. And at the same time, they create a gradient. Okay, that gradient is the one that will facilitate the absorption of these sugar monomers. But on the basolateral side, we also have transporters which allow facilitated diffusion because we are moving from where they are high in concentration to where they are low in concentration. But these sugars cannot pass through the cell membrane. They have to pass through these channels or through these transporters. For that reason, this is called a facilitated diffusion. Okay? So in this manner, that's where you end up having a lot of these glucose or galactose or fructose being absorbed. So there is something else I wanted to mention about. There is the monomer, there is the enzyme lactase, which is going to break down the lactose to glucose and galactose. Now, you find that in a newborn, you really have a lot of this enzyme being produced in the brush border. And the reason being, newborns rely on milk. So they need energy coming from the milk. So because they are so dependent on milk, it will be breaking down the milk sugar to provide energy. However, as these individuals keep growing up, as they become less dependent on milk, you find that they produce less and less enzyme, lactase enzyme. And you find that in some adults, they actually lose even their ability to digest the milk sugar. And those are the people we start saying that they have lactose intolerance. Why? Because they have reached a point whereby they are not producing this lactase enzyme. So you find that in adults, generally, it's very little in amount. And as you take in your milk, it's supposed to digest. And you're supposed to be okay. Now, the problem is, at times, if you are having lactose intolerance, it means that you are not producing this enzyme. If you don't produce this enzyme, then what happens is that this lactose is not digested. If it is not digested in the small intestines, then it cannot be absorbed. Then what happens is that it will be passed down the digestive tract and it will end up in the colon. Now, who remembers what is rich in the colon that causes digestion? Yes, Asna. Unmute yourself.
Is it the bacteria?
You have a lot of bacteria. And now you are providing this bacteria in the colon with a lot of lactose. So now they have a lot of sugar to work on. Unfortunately, in the large intestine, we do not have oxygen. So even the breakdown of these products, of this sugar, is usually anaerobic. And they end up producing lactic acid. Now, lactic acid has several things. One, it will cause water to be absorbed, a lot of water to be taken into the lumen to try and neutralize. Alternatively, during this anaerobic breakdown, we also produce a lot of gas. So people with lactose intolerance are likely to have a lot of watery stool, and secondly, they are going to experience a lot of flatulence, whereby you are just releasing air in the other direction. So you find such people, because of lactose intolerance, they feel bloated. They have a lot of gas in their digestive system. So you take, after taking things like milk, they'll end up having bloating, and they'll end up producing a lot of gas from the GIT. So normally the such people are just told you really have to limit things that are a source of lactose. So I thought that it's good to mention that, because that is the end product when you don't have a total digestion of the carbohydrates that you ingest. Now, with that, I think the next thing is I go to protein digestion. Yes, Samuel.
Hello, Prof. What did you say is the effect of lactic acid on the absorption of water? I didn't get that one.
Now, in the digestive tract, because of this acidity, normally in the colon, we usually have absorption of water, isn't it?
Yes.
We absorb water. But in this case, because you are producing this content of acid, and remember, with acid, you are also trying to produce bicarbonates to neutralize the acids. So in the colon, you are having a lot of bicarbonate production, but now you have excess acid production. So what you are trying to do is to try and neutralize the environment. You end up having, instead of water being reabsorbed, you end up drawing water into the lumen of the colon. Are we together? So the end effect is that where you are supposed to dehydrate the stool so that you end up having the semi-solid pieces, because of this drawing in of water, you end up having watery stool, so that the pieces are very watery. Is that okay now?
Yes.
Okay, so now let's go to protein digestion. Now, the source of proteins is mainly dietary, but you find that apart from that, I told you that we have enzymes that you produce are proteins. They are also found in the lumen. So, and in the lumen, we have digestive enzymes for proteins. So even those ones will act as a substrate for the enzymes that are found within the lumen. Secondly, you know that the dead tissue will have them a lot of protein products. So whether it is the connective tissue layers, whether it is parts of the intestines, the cells that have proteins embedded in their cell membranes, all those are producing or they are contributing to the proteins that end up in the lumen. And these become a substrate for the enzymes. Now, protein digestion normally starts in the stomach. In the stomach, we have pepsin, but it is produced in the form of pepsinogen. But in addition, we also have the milk protein, casinogen, which is also digested by renin, also produced in infants. So in infants, we have renin to break down the casinogen. That is also a milk protein. Then we also have lactase to break down the lactose. But these end up becoming minimized as the person or the individual grows or matures. So as the digestive tract matures, the level of these enzymes drop. So the main one we rely on is the pepsin, which acts like the big chopper. It's an endopeptidase. It will keep on chopping the long peptide for the peptide chains into smaller or shorter for the peptide chains. Now, these peptide chains, they end up coming from the stomach. Remember how this is happening within the stomach. So when you leave the stomach, you go to the duodenum, and it is within the duodenum that you have the pancreatic enzymes. The pancreatic enzymes that will target proteins are the trypsin, which comes in form of, all of them are zymogens by the way. Trypsinogen, amitripsinogen, procarboxylase. And what will happen? Remember, at this level, we do not have the aminopeptidases because the aminopeptidases are usually the brush border enzymes. But we have carboxypeptidases. So if you remember the difference between the two. Yes, Anastasia?
Yeah, I was asking about renin. You've said it digests soluble casein into insoluble, or carcinogen into insoluble casein. So that insoluble casein is the one that will be absorbed, or should it not be soluble for it to be absorbed?
Now, yes, I think that one I did not capture it properly. Yes, I think it should be that way. From the soluble to the, but you are actually going to digest it further. So when you have the casein, it will be digested further to be broken down. So if once it is able to be attached by to be digested by the other proteins which are within the duodenum. So yes, I think it is coming from the soluble to the insoluble. So it's the other way around. I'll come to that later.
Okay.
I'll check on that later. Yeah? So when it comes to these carboxypeptidases versus the aminopeptidases, you find that these ones are found, they are actually the pancreatic enzymes, the other ones are the brush border enzymes. All of them will start breaking the short peptide chains into individual amino acids. The amino acids are the ones which can actually be absorbed. However, at times you find that we also have some short polypeptides which are absorbed wholly as they are. At the brush border enzyme, we release individual amino acids. Or at times you can also have the dipeptides. Instead of having one amino acid, you can have just individual monomers attached by the peptide bonds. So these are the ones which will normally be absorbed. They'll be absorbed through the epithelial cells via the brush border enzyme. They're the ones which are going to use the transport carriers. But there are some other cases whereby we may have short peptides rather than the dipeptidases. The dipeptides, we may have short chains and they are absorbed wholly as they are. So if you remember, we mentioned some time back a transport method whereby the whole protein is translocated as it is into the cell, and then it is passed into the basolateral side as it is. Who can remember what the process was? Whereby you have the cell membrane and you have proteins on this side. But then what will happen is that this protein, if I use the black, it will be sort of like engulfed, will be taken in, and the whole of this unit with the protein will be moved to the basolateral side where it will be released. And I remember mentioning that type of transport method in class. What did you call it?
Is it endocytosis?
Okay, there is the endocytosis. Usually endocytosis, they end up inside the cell. So it is a form of endocytosis, but with endocytosis, once you have inside the cell, then usually they are disintegrated or they are lysed by the lysozymes or something happens. So Nicholas, your hand was up.
Yeah, I wanted to say phagocytosis.
Even phagocytosis, it's the same. So it is a method. It's actually like a phagocytosis. Yes, Eden?
Pinocytosis?
Okay, Pino. With the phago, it's taking large components or material. Pino usually does not take the solid materials. It also engulfs some liquid portions, causes pinocytosis. Yes, Axilla?
Is it the transcytosis?
That is, yeah, it is transcytosis. Okay, it is transcytosis. It is transcytosis. I remember during the time when we were discussing the transport methods, the different transport methods, the only two I mentioned, actually there were three. I said phagocytosis, pinocytosis, endocytosis. But later, I think it was during the pre-lab, I mentioned transcytosis. And that's why I was telling you at times it's good to write things down. Because this one, the transcytosis, the transcytosis, I mentioned it, I think, during the pre-lab. You recall that? So it's only one person who can recall that one. But transcytosis is when you take the whole of these protein components and you transport it as it is, you translocate it into the other side, the basolateral. This type of transportation or absorption is important, especially in very young animals or very young infants. The minute you're born, you hardly have antibodies. You rely on your maternal antibodies, but the maternal antibodies are coming to you through the milk. Now, if you take the milk and the antibodies are mainly proteins, all the immunoglobulins and all that, they are proteins. So the infant, if they ingest those proteins and they start breaking them down, it means that they will not have any immunity to them. So what happens is that these proteins are engulfed, whole as they are, and they are translocated to the other side so that they can provide the child with immunity. So that kind of transport mechanism is important in infants so that they can also provide them with immunity. So the other ones which are broken down and they end up releasing amino acids, how are they transported? So if you understood the transport mechanism for the glucose, then it is very easy to understand that one of amino acid because it is very similar. So I think this is a diagram I had shown you in the pre-lab, showing you the digestion in the stomach, cutting it down to short polypeptide chains which are absorbed within the intestines. So this diagram is actually showing you the mechanism of transcytosis, whereby you have some of those proteins and they'll end up being they attach to some receptors which will initiate the engulfing, and once they are engulfed like that, they are transported to the other side and they are released. So that is just one of the ways. However, there is also what you call the paracellular transport, where it passes through between the epithelial cells. So I had not mentioned that one, but you can also have when it passes through the between the epithelial cells. So, like I said, you understood with the what is it called? With the amino acids, it's similarly transported like the glucose, where we have the protein, the sodium potassium pumps create a gradient, allowing sodium to flow in. As sodium goes in, it also moves in with amino acids. And then these amino acids will use the amino acid transporter on the basolateral side, and it moves into the blood capillary system. Then from there, of course, it is in the blood system. Now, one of the things they have not shown here is the proton pumps. At times, we also have pumps that will allow hydrogen ions to be released in exchange for amino acids. And then when you have the hydrogen ions, what you're doing is you're creating an environment which will also allow water to move. And then the funny thing is that, not the funny thing, usually when you have the sodium moving in, it also creates tonicity, which will also cause the water to go back in. So it is also acting as a form of water absorption. But this is usually provided by the proton pumps, and the proton pumps, they are also exchanging with them, they are transporting together with amino acids. So those are two methods of transporting proteins. I just showing you one here, but you can also use the proton pump. So I think the next one, or this diagram is just showing you a summary of those two, where you have the luminous site, and then you also have the basolateral site. So it's showing you the breakdown of proteins and carbohydrates into individual monomers, and these ones they are transported and they end up in the capillaries, which end up in the hepatic system. So the one type of food we have not discussed here are the lipids. So although we say that we have the lingual lipases and we have gastric lipases, we find that the amount of digestion there is very, very minimal. It doesn't even account for any of the we will say it's a negligible amount for the ones that ends up in your system. Most of the digestion is from the lipids that you actually ingest. It occurs through the pancreatic lipase. The problem is, because they are lipids, they are not water soluble. If they are not water soluble, then it becomes very difficult for the lipase, because lipase is an enzyme. And the lipase enzyme is dissolved in the aqueous medium. And the aqueous medium does not interact with the lipids. So for the lipase to have access to the lipids, you need to break these lipids into smaller components. The problem is, if you have a large droplet of lipids, you have such a large droplet, or if it is a fat globule, it's just a fat solid matter, then it is very difficult, because the only enzyme that can access is on the surface. So you find all these components in the middle cannot be digested by the lipase enzyme. So the amount that is free for absorption will be very little. Now, to enable lipase to access the lipids, this droplet must be emulsified. So you must bring in the bile salts. And if you remember, the bile salts are coming in through the bile, and the bile is released in the duodenum. So the fat will have passed from the mouth, minimal digestion, into the stomach, minimal digestion. Then it goes into the duodenum. In the duodenum, it is exposed to bile because we have bile coming from the hepatopancreatic duct. So the bile will act like a detergent. Remember, the bile is amphipathic in that it has both hydrophilic and hydrophobic ends. So what will happen is the bile will come and the hydrophilic part will be exposed to the surface, and the hydrophobic part will insert themselves in between the fat layers. And remember, all this is happening on the surface. Now, in the duodenum or in the stomach, there is a lot of mixing and churning. So things are hitting against the wall. And what is likely to happen is that as you feed these droplets, it will be broken down into smaller droplets. But remember, these droplets had the bile salts on their surface. So now what will happen is that the bile salts, because they are on the surface here, they will not allow the droplets to coalesce again to form the big droplet. So it will keep these small droplets separate. And continued mixing and churning will break these small droplets into tinier pieces. And when they become smaller again, they are surrounded by the bile salts which keep them separate. Now what happens is that now the enzyme lipase will be able to come and act on these tiny lipid molecules here. And it will be able to start releasing fatty acid chains. So if you look at the green, or let's use black because that's what we had. So what it will end up doing is releasing fatty acid chains, free fatty acids. At the same time, it may have some dry acid glycerols. That means it has two fatty acid chains. And you have the triacyl glycerols, which are or triglycerides, which can be broken down again by the lipase to release these components. And of course, you have glycerol. Glycerol will pass through the cell membrane very easily. It has no problem. But once it is bound to a lipid chain, then this cannot pass through the cell membrane. So they have to devise a way of allowing these to pass through the membrane. So now we have understood what happens when we expose the fat or the lipids to the bile salts. It's the breaking them down into smaller droplets so that the lipase can act on them. So my question is this chemical or mechanical digestion?
Mechanical.
So what you are having, because there is no breakdown of any bonds here, then what you have here, emulsification is just like mechanical digestion. It is making them smaller. Now, once you make the the mechanical digestion, you expose them to lipase, and that allows the chemical digestion to take place. So this is a flow chart explaining, and I think I had already shown you this during the pre-lab. how food is ingested. The lipids are ingested in the mouth. You have only lipase, gastric lipase, accounting for about 1% of digestion. The rest ends up in the small intestine, and because of bile emulsification, they end up being exposed to pancreatic enzyme, and they end up releasing either the fatty acid chains, which you are calling the monoglycerides, and then you can have the diglycerides, or you can have either free fatty acids or glycerol with a fatty acid, which is the monoglyceride, or you can have glycerol with 2 fatty acids, which you call them the diglycerides, because when you have the glycerol with 3 fatty acids, then we are talking of the triglycerides. So after lipid digestion, these are the products that we are likely to have: 3 fatty acids, monoglycerides, and the diglycerides. Now, it is good to note that the products of these fatty acids, they end up being absorbed in different ways. We may have triglycerides which have very short fatty acid chains. So when these ones are broken down, or when they undergo lipase digestion, you have very short fatty acid chains. These fatty acid chains, which are very short, are able to pass through without any modification. They pass through the cell membrane and they end up in the blood circulatory system, and you'll see how. But then those that are longer then have to go through some modification to allow them to be taken up. So how are they taken up? First, they'll have to get into the enterocytes, and then from the enterocytes, they are modified so that they can be excreted from the basolateral side. So I think I'm usually better with the diagram, so I got this diagram to explain what is happening here. So you have the lipids, which are triglycerides, and they are being broken down to release either the monoglycerides, diglycerides, or free fatty acid chains. If it is glycerol has no problem. It will pass through. At the same time, if we have the short fatty acids, even those ones will end up passing through the cell, the enterocyte, and they end up in the blood capillary. The problem is when you start having them getting into the cytosol, and from there they undergo what we call reesterification, whereby they go back and reunite with glycerol. When they reunite, they'll end up having a triglyceride. Now the triglyceride, as it is, cannot enter the blood circulatory system. So what happens is when you end up having these reesterified triglycerides, they'll have to be modified so that they are packaged with protein components. So proteins have to come and wrap around these triglycerides and any other lipid soluble component. For instance, you may have things like cholesterol, and what they'll end up doing is when you have a mixture of lipids and cholesterol and all these proteins are products of, say, the phospholipids, they'll end up combining with proteins and they form a unit called the chylomicron. This chylomicron is actually exocytosed, and once it ends up, it goes into the lacteals rather than into the blood capillaries. So what you'll have done, you'll have used the fatty acids, broken down But before they get into the enterocyte, they form what we call a micelle. A micelle is when you have these free fatty acids, they usually have the polar heads on this side, and then you have the fatty acid chains. These heads will mix with water very easily, but the fatty acid chains will hide. So what happens is that if we have several of them and they are hiding all their tails, you'll end up having something spherical with the polar heads on the surface and the chains towards the inside. So when you have this unit, it's what we call the micelle. And the micelle is able to move through the mucus lining and go all the way to the brush border. And then from the brush border is when it starts to release the free fatty acids. So the micelle helps in transporting these chains that are formed through the mucus lining to expose them to the brush border, where once they release the individual fatty acids, they are able to diffuse through the cell membrane and they end up within the cytosol. So within the cytosol, you'll have all these fatty acids will now become reesterified, and then once they are reesterified, they'll end up combining with proteins to form the chylomicrons, which are then released into the lactease. For that reason, you find that individual triglycerides or cholesterol molecules, they'll usually end up in the lymphatic system. On the other hand, if you have any free fatty acid chains, they can also end up here, but most of them can also end up in the hepatic portal vein. And that's why when you start checking in the hepatic portal vein, you are not likely to find triglycerides, but you can find those short fatty acids. And they have to be the short, not the long ones. The long ones have to go through the esterification. So in this manner, you'll have absorbed the lipids. Yes, Lucy?
Okay, so I have a question.
Yes.
So the micelles, they basically just help to transport the 3 fatty acids. Do they like just dump it, or I don't know, they help them enter through the layer or what? I don't get that part.
You don't get that part. I think I have a better diagram. Let me show you the next diagram. Another one, which are the mucus layer. Okay, I'll use this one here. So I'll draw my mucus layer. So when you have the large fat droplet, then it comes in contact with the bile salts. It will break them into smaller droplets. Then you'll have now the lipase coming in. And the lipase, remember all this is happening in the lumen. And the lumen has aqueous medium. And now when you start releasing the aqueous media, so we have the mucus layer around here, just at the brush border. So now, because you have this particular, it's an aqueous layer, you have released fatty acids. The fatty acids, then what happens? You have now those long chains. And they want to hide from the aqueous medium. They start to form the micelles. And once they form the micelle, they have these individual layers here. So now what will happen is that this will be able to pass from the aqueous media, even the mucus is more aqueous. So it is able to stabilize and to move all the way to the brush border. Now, at the brush border, what will happen is that they'll start to release the fatty acids, which will end up moving or dissolving past here. So what it is doing, it is helping the fatty acids to move across this aqueous medium and reach the brush border. The reason I can come up with is, if you have a glass of water and you add oil, oil will stay on the surface and the water will remain at the bottom. Now imagine at this point is where you have the brush border. How will this oil or fatty acids be able to be transported to reach this place? So the micelles are acting in a way because they are hidden and they are able to cross through this place. Remember, some of them are still attached to the bile salts and they are just moving. As you digest, you release the fatty acids. If they are free fatty acids like this, they hardly reach the brush border. But when you aggregate them together to form micelles, the micelles are able to reach the brush border. And when they reach the brush border, they actually start releasing the individual fatty acids. So it is just for the micelles are in a way so that they can be able to dissolve or to be dispersed within this aqueous media, which is not possible when they are within the lumen. So if they were to be digested in the lumen and they remain as free fatty acids, they'll never be able to reach the brush border. So they aggregate as micelles so that they can be able to reach there. Once they hit the wall, they are trapped there, and once they are trapped, they can release the free fatty acids. Are we together?
Yeah.
Or have I confused you further? Remember, remember that this brush border or these epithelial cells, if this is a cross section of the small intestine and we have the villi, okay, and the brush border is here, okay, it is just at this point. Now imagine the digestion is taking place here and you have all these free fatty acids. If they do not reach the brush border, there is no way they can cross. The brush border has these finger-like projections. So every time you are squeezing, you are pushing some of these components all the way to the brush border. The fact that you have micelles, those micelles will be able to reach those microvilli. And once they are attached there, they can be able to release the components. But if they are not there, then the free fatty acid will just float and they will float back. So they are helping in holding the free fatty acids to expose them to the brush border so that they can be able to pass through the cell membrane. Okay.
Yeah.
I hope that is clear.
It is.
All right. Thank you. So the other food components that we absorb are usually the nucleic acids. The nucleic acids, they are made of three components. We have the ribose sugars, we have the phosphate group, and we have the nucleotides. Give me Okay. So when it comes to the nucleic acid, you have the nucleases. They are the ones which will end up breaking these down into the individual monomers. So of course, when you have the pentose sugars, it's just like the simple sugar, it will be absorbed. But then when you have these other ones, they'll have to be digested by the nucleases, and they'll have to break them down to allow them to pass through. The phosphate groups are just like any other ion, so they are able to pass through, just like they are absorbed, they are utilized just like any other phosphates. They are taken up by the phospholipid bilayer and they are able to be transported across. So the other things that we have being absorbed are the electrolytes. And all of them, except for these two, calcium and iron, these are absorbed. Most of the electrolytes will be absorbed down the concentration gradient. But for calcium and iron, these ones, you need a transport mechanism whereby with calcium, you require vitamin D to facilitate absorption. And most of the water, as we said, it always follows the electrolytes. Most of the water will follow the sodium chloride. Sodium will be absorbed with all those transport mechanisms that we are mentioning. They are concentrating the sodium to the basolateral side. As you concentrate the sodium, you also follow it up with the chloride ions. So what you end up doing is increasing tonicity, water follows. So you usually find that as you absorb the nutrients, you also absorb the water components. Most of these other electrolytes will also be absorbed in a similar manner. Now, if you look at, I don't know whether I have that diagram, which is showing you all the different parts where you have absorption. Yeah, again, I missed that diagram. I have a diagram that was showing you the different parts of the GIT and the different components that are absorbed. Now, you find that most of the absorption will happen within the small intestine. And this will happen for all the nutrients that we're talking about, from the amino acids, from the monosaccharides, the lipids. Most of it will happen in the small intestine. However, the two that I have mentioned, we have calcium and iron, will happen at the duodenum. Number two, vitamin K will end up being absorbed in the large intestine. And I say this because this is usually released within the large intestine, the colon. Now, in addition, we also have the absorption of the bile salts. So remember the biles, they are being released by or they're being synthesized at the liver. Once they are synthesized, they are released into the lumen. And they are transported. As they go down, they keep on emulsifying the fat and they allow the lipid digestion to occur. However, you do not want to keep on resynthesizing the bile salts. So most of the bile salts will be reabsorbed back into the blood circulatory system. But this reabsorption usually happens at the terminal portion of the ileum. So I'm not sure whether I have it in this diagram. Let me just see if I have it here. No, I don't. But I think it is in the diagram that I have uploaded. The slides that I have uploaded on Blackboard, they will show you how bile salts are reabsorbed back into the system. And this usually happens at the terminal portion of the ileum. Most of them, about 95%, will be reabsorbed. Only 5% ends up within the colon. And of course, it is excreted out together with the feces. Now, bile salts, if you look at the way they are synthesized, some of them are synthesized from breakdown of red blood cells, hemoglobin, and all that. So their color is usually a dark brownish color to red. For that reason, most of these bile salts will end up having a very dark brownish color. And that is normally the color that you're going to see in the stools because of the 5% of the bile salts that are excreted out together with the feces. Now, if you have a problem with the liver, it means that one of the functions, which is to manufacture or to synthesize the bile salts, then you're going to have a problem in producing bile salts and bile pigments. It means as your feces pass through the digestive tract, you're not going to have those salts, the 5%, they will not be there. If they are not there, it means that the kind of stool you are going to release will be very pale. Some of it appears between yellow and a very light tan color. So one of the ways of telling if you have a liver problem is by the color of the stool. If it becomes very, very pale, almost whitish to yellowish, very light yellow, it means that you do not have any bile pigment. It means that you have a problem. Now, the other thing that you end up seeing is because there is no breakdown of those products, you'll end up having an accumulation of breakdown products of the red blood cells. Some of these is things like the bilirubin, biliverdin, all those things, they have that color, the yellowish, very dark yellow to reddish color or to brownish color. So if you have a buildup of those products, which are the ones that are normally used as the raw materials for synthesizing the bile salts, if you have a buildup of those, then they'll end up showing in all your light membranes where you have your membranes or places like in your eyes where the whites of the eyes are supposed to be. They start appearing yellowish in color. If you look at your fingertips, which are normally pinkish because they are showing the red color of the blood, they start to appear yellowish. If you look at your skin surface, it also starts to appear yellowish. All this is because of the buildup of these bile pigments. So in addition to synthesizing and helping in digestion, it also helps in the breakdown. In eliminating the breakdown of these blood components. So I think with that, I have explained the symptoms that you're going to see. But what is that condition called? Who has ever heard of that condition? What is it called? Ever heard of jaundice?
Yes.
So that is the situation whereby when you have all this buildup of pigments, they start showing yellowish color in all over your body, and especially in the whites of the eyes, the fingertips, and when you check inside the mouth, it's appearing yellowish, then you say you are jaundiced. Then it is indicating you have a problem with the liver. So I think with that, I say today it will be a long lecture, although it's still going past 12. I think I have captured most of the digestion, absorption. We have done secretions, we have done regulation. So I think we are ready now to start with the respiratory tract. Any questions so far? Okay, so with that I take attendance. I've been talking the whole morning, so my voice is really hoarse.
Oh, madam, I wanted to ask if you have uploaded the notes on migrating water complex.
I thought I had. I had enabled all the notes to be seen.
Okay, that's all.
Yeah.
Thank you.
Okay. So I'll take attendance now.